In this video, we will discuss about how they find out that the cGAS or sting pathways can also induce BH3 only genes by BH3 mimetics. So from the first figure that we have present, understand that they have seen that this uh, not just P53 pathways get upregulated when they inhibit this pro-survival proteins, but also this NF-kappa beta pathways are also upregulated. And these NF-kappa beta pathways are mainly upregulated by these STING pathways. And one more thing that they have found out that in the case of, they have observed that in the case of TRP53 knockout, there is a reduction in the BH3, there is a reduction in this P53 effector genes, but not completely abrogated. That can be seen in the P53 knockout for this BAME or this Puma. So from this they come to a point that there could be some other pathways that is also upregulating P53 effector genes. And from this gene set enrichment analysis, they think that the pathways that are upregulated by NF-kappa beta or this interferon could plays a role towards upregulating these P53 effector genes. So when they did the literature analysis, they find out that STING will also upregulate this NF-kappa beta pathways. Uh, so therefore, from this they come to a hypothesis that it's the these STING pathways could possibly upregulate these P53 effector genes. So in order to validate that thing, they take this T53 knockout and check the effect, check the phosphorylated P stings and phosphorylated TBK1, which is the markers for the cGAS or sting pathways activations. And what they have find out is that when they use these BH3 mimetics or when they suppress the pro-survival genes, this is it after around 4-6 hours. In the case of control, they see the phosphorylated sting or phosphorylated TBK1, and similar results they have seen in this TP53 knockout. And when they did the same experiments on these DHL cell lines, they also see the same results. That is, phosphorylated sting and phosphorylated TBK1s are upregulated, and also this IRF3 is also upregulated. Thereafter, they did the analysis on the sting knockout cell lines. Here, they do not see any stings as expected, as the sting pathways are being knocked out. Thereafter, what they did is they check in 3 different cell lines, and also they check in the TP53 knockout and sting knockout the effect of these P53 effector genes. And the IRF7 is the gene that is responsible for sting pathways activation. So first we will discuss about the P53 effector genes, that is NOX, PUMA, BIM3, and we can see that in all the 3 cell lines, all the 3 genes, they NOX, PUMA and BIM3 are dysregulated or downregulated variably. In some cases, there is no downregulation occurs, such as TF3 knockout and the beam pathways. So this could be because the other pathways, that is sting pathways, might be able to compensate it better. And when they check the activation of sting pathways using the genes that are triggered for activating the sting pathways, such as IRF7, they see that in the TP53 knockout, there is activations of sting pathways in all the 3 cell lines. So all this indicates that in the absence of TP53 or TP53 deficient cells, sting pathways might try to compensate its absence. So, and one more thing that I forgot to mention you, this experiment was done with this QED OPH, that is caspase inhibitor. As from the literature, it is found that caspase can inhibit these sting pathways. That is the reason in normal case they are not able to see these sting pathways activations, as the apoptosis by MOMP will trigger this caspase formation and the caspase will inhibit the sting pathways. So these panel D experiments and panel ABC experiments are done with this inhibition of this caspase pathway, and the experiment were also done without this QED OPH, and those results are shown in the supplemental figures. So from this, the final conclusion that they come that whenever this BH3 mimetics occurs, then it will trigger this MOMP, and this MOMP will trigger the activation of STING pathways, and that will cause the further upregulation of this BH3 only proteins that will suppress pro-survival proteins. But there is a competitions that has been observed between these caspase formations and these STING pathways formations, and those results are in supplemental figures. So I hope you understood how they find a hypothesis, and then did some experiment to validate their hypothesis that STING pathways can also induce BH3 only genes by BH3 mimetics in the absence of p53. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.